The topic of the talk here is autism and gene therapy. And I've been working on ideas for gene therapy for a long time. And I would like to tell you, I mean, what is the status, what we are doing for that. And also take this opportunity to really set the stage on what is gene therapy and why we should care about gene therapy and autism. This is because I feel that sometimes my colleagues working on gene therapy, when they present or when they talk, I think they're often misunderstood, even by the community of autistic individuals. And again, because there is no contextualization of what the scientists are trying to do here. So I decided to divide my talk in different topics. But before I move on, I just would like to say that I'm a faculty here at UCTV. I'm also the director of the stem cells. You are in a place where human stem cells are taking place. The labs are in that building. There are people from my lab here. If you meet one of them, you are welcome to ask them to tour the lab. So you can go and check it out. My lab manager is around. She can organize those things. But also most importantly on my CV is that I'm a father of Ivan, and some of you know him. So Ivan is an autistic boy with 16 years old now. And so most of my motivation is actually because of him, because we wake up together, we interact, and I recharge my energies because of him. So if I'm full of energy, it's because I've been around him for so long now. All right. So the way I decided to structure this talk is in 5 steps. The first one is, as I started to say, to contextualize the autism spectrum and exactly what we are trying to do here. And then I want to provide a very clear definition of gene therapy. I still think that there are confusions out there. It is not a new concept for science, but it's becoming kind of a mainstream in society. So make sure that everybody understands. And then I'll introduce the human model. I mean, why we use a human model to study autism. I'll show you the core of the science, which is the proof of principle that a gene therapy approach might work and why we are excited about that. And then I'll talk the next steps, what we are doing and what I think needs to be done as a next step for these to become successful and move forward. All right, let's start with put that in a context. And the reason I'm doing that, and for those who actually watch the videos from the conference last year, might remember that we start with a conversation with a scientist who had his research shut off because of the lack of interaction with the community of autistic individuals in his surroundings. So they didn't understand why he was studying the genetics of autism, and the work was temporarily shut off. And thankfully, I mean, everything was clarified and he's back on track. But we took that opportunity to have him talk and discuss here with our community, I mean, about that experience. So I'm good on our community because we have been doing this for several years. So I think we interact really well with each other. So I'm not too worried. But because we have a hybrid event now, and this is being broadcast worldwide, I'd rather, again, I mean, set the stage for what I'm going to say or talk about gene therapy and autism. And first, by contextualize autism spectrum disorders, this is what most, I would say, neuroscientists and medical doctors will characterize as autism spectrum disorders. You have different levels of autism. High-functioning autism would be what we call level 1, where these individuals will need some assistance. Some are already fully independent, so they don't really need too much more than that. And then you have in the middle, which would be level 2, where they might suffer from some comorbidities or some consequences of autism and might require a little bit more of support. And then finally, we have level 3, where these individuals will definitely need some support, sometimes one-on-one, sometimes 2-to-1, to have that individual functioning. So yeah, it's a large spectrum, and over the years the spectrum has grown a lot. And I honestly think that in the beginning I thought that this would be good because we have more representation. Now I think that we need to divide it again, and I think there are discussions about how to do that, but that's not the topic here. So we have to work with these right now. So again, I mean, this is what most medical doctors and scientists will see. What we see in societies is slightly different, would be something like that. You have perhaps in one extreme of the spectrum, what we call the high functioning would be the Aspergers. And these are often what you see in movies, in TV shows. They're often called smart kids, genius, or they're just different. Some of them are completely independent. Some of them still requires like some help. Most of those are looking for a job, or they already have a job. They just don't function well on that job. And there are a vocal part of that fights for neurodiversity, and that's amazing. It is actually amazing to see how that community has grown up to fight for what they really want. And we've seen here at the TPF, and we help that voice to elevate as well. But on the other side, you have what we are now calling, or some people are advocating to call what would be profound autism. And that's highly different. That's invisible to the mainstream society. So you don't see those kids most of the time because they don't go out. They stay at home. It's very hard to leave the home with someone who is severely affected. So these are sometimes syndromic. They might have like a syndrome that are under the umbrella of autism. They require assistance 24/7. They will die if you leave them alone for 1 hour. So they have no idea what's a job. They have no idea what's diversity. They're not part of any conversation. So, and there are some data saying that these are like 50 and 50% of the whole spectrum. Debatable if those numbers are right, but nonetheless, what's important here is that the extremes are getting very opposed to each other, to the point that what one end of the spectrum is fighting for example, the Aspergers, inclusion, diversity, acceptance, they would even disapprove words like patient, disability, intervention, cure. Come on, I don't need that. And again, I mean, it was beautiful to see how this was growing over the years, and the rise of the neurodiversity among autism, something that we've been talking on these conferences for a long time. However, we need to be aware that the profound autism population cannot fight for anything because they cannot even talk. So most of those kids are nonverbal. They cannot. They have like severe neurodevelopmental disorder. So most of the fights are actually done by the families or the parents who are, again, I mean, supporting them 24/7. So the time that they have to fight for something is really minor. So it's a very sensitive population that it's perhaps the most vulnerable among the spectrum are the ones with profound autism. And of course, I mean, these are actually patients. They do have disabilities. They need intervention, and their families are actually hoping for a cure. So there is nothing wrong about what the two extremes want to fight for, the two extremes want for. We just need to understand that they are actually two extremes of the same spectrum, something that over the years it seems that we are losing the sense of what are the needs for those two, which, to be honest, they are not mutually exclusive, because the profound autism will like to have inclusion, diversity, and acceptance at the same time. So not mutually exclusive, so we should not put them against each other, but we should converge them, I mean, making them work together, which would be even better. So what I want to point it out is that for the topic of what I'm proposing here, which is gene therapy, I'm not talking about Asperger's, I'm not talking about high-functioning autism. I'm talking about profound autism, people that will die in 1 hour without supervision. So these are the ones that I'm talking about. So gene therapy, it's for those. So if you are not in that part of the spectrum, you don't need to worry about that because we're not studying you, we're not providing any therapy for you. We are doing for the other ones. So that's the contextualization. So let's move on to what is then gene therapy, why we are doing that. So this is my definition of gene therapy, a way to overcome the consequences of a genetic mutation in a cell, hopefully restoring the proper function of the cell, tissue, and or behavior. Basically, we're trying to correct something that you are born with, some genetic mutations, and hoping to restore the proper function of the cell tissue or the brain. So how does gene therapy work? So for those who are not scientists, you might ask, I mean, how do you do that? I mean, you have about 86 billions of neurons in your brain that controls all your emotions, your behaviors, the way you talk, the way you present yourself. These are all orchestrated by 86 billions of neurons and about the same number of known neuronal cells that helps the brain cells to orchestrate as well. So how do we fix a mutation in each one of these? So that's not an easy task. And this is how gene therapy works. So virtually what scientists can do is to pack the right version of the gene or enzymes that can fix the mutation of a specific gene inside a virus. And we use virus because viruses have a good way, they're very efficient on penetrating human cells. I mean, after the pandemic, we all know that this is true now. And we use a virus that does not cause any harm to the person, and it's called AAV, an adeno-associated virus. So this is like a very mild flu virus. So we remove all the genome of that virus and we place these enzymes or the correct version of the gene inside that virus. And then we use the virus to deliver the correct gene or the enzymes that will fix the mutation back into the person. Those viruses would migrate to the nervous system and will deliver the correct copy of the gene to your brain cells, right? So in concept, that's amazing, that's perfect. But practically, we still have like a long way to go because there are several problems with this idea. Your body might react to the virus, your body might react to the correct copy of the gene, because remember, if you have a mutation, maybe your cells, your immune cells, never seen the correct version of the gene, so they might fight it back. And the delivery part is the most problematic. The virus does not fully penetrate the human brain. It would infect only a few cells. It's not 100% correction. And sometimes there are doses problems. Some genes cannot be you cannot have like too much of the gene in there, you cannot have too low of the gene in there. So you have to control all of those aspects. So there's still like a fine-tuning problem with this technology before it becomes like something that you can move on into clinics. Although there are several clinical trials and protocols already moving on. I mean, if you go to clinicaltrials.gov and just place in a search like gene therapy, you're going to see hundreds of gene therapy clinical trials going on. So this is definitely moving to mainstream. It's not perfect, but it's the best what we can do right now. And so, I mean, it's a good tool. All right. Okay, so yeah, why we're not doing that for autism? We should be doing this for profound autism or the ones that we know the genetic mutation right away. Well, there's a problem that scientists have when we are trying to study autism, which is the lack of a model. And especially if you take like syndromic autism, profound autism, these are mutations in genes that affect your neurodevelopment. And meaning that you are actually born with autism. You don't acquire autism through your life. So your autism actually starts in utero. So how can we actually study the brain of autistic individuals when they are forming in uterus without use of non-invasive technology. So we cannot do that. Instead, what scientists have been relied upon was in different models, such as postmortem tissues. I mean, people with autism eventually die. Some families donate the brains to research, and we have a couple of those, so we can study. But the person is already dead. So the tissue is not alive, and the damage or the alterations is already formed. I mean, we don't capture what happens before, during the formation of the brain. Another way to study autism is to collect blood from people. But blood cells are not brain cells. Blood cells does not make connections, does not have synapses, does not influences your behavior. So there is so much you can do with blood, and most of the genetics of autism was done in blood. So it's still a very useful model, but doesn't get to the brain. And then finally, we have animal models. Animal models are tricky because you can create mutations similar to the ones that you see in patients in a mouse model. And the mouse model might seem autistic, but sometimes it's tricky because the mouse brain and the human brain, they evolve in different ways. What a mouse might be doing that seems like autistic, for example, having like a repetitive behavior, might have nothing to do with the circuitry or the brain cells that actually do the same thing in humans. So most of the times, especially when we are moving into treatment, mouse models have failed, miserable, especially for the brain. So you can see as a scientist, we don't have too much options here. And that's why I decided to bring, or me and others in this field, to take advantage of stem cells, human stem cells. Because with human stem cells, I can actually make brain stem cells. Stem cells are the ones that can differentiate or specialize in different brain tissues, including brain cells. And it was in 2008 when a researcher called Yoshiki Sasai from Japan showed us that if you use a specific combination of factors, you can guide these cells to form brain tissues in 3 dimensions. And this was the first publication. 2008 was when I was starting my lab, and I saw that. I said, oh my gosh, that's the model I need for autism. So I started following his path and improving protocols that now we have, way more robust than what he previously did. And so we could derive stem cells from autistic individuals and controls and compare them. So I can compare directly their brain cells in a dish, in a controlled situation of the lab. So there are different ways of doing that. These are the different protocols that we have from the stem cells. You can create brain organoids that have multiple brain regions, or you can guide them to specific brain regions if you know where are the brain regions. For example, in autism, we focus a lot of attention in the prefrontal cortex because we know it's a part that is affected in autism, and it's a part related to human cognition, and lots of processing of information happens in the cortex. So we now have tools to guide the stem cells to become cortex, a brain organoid, cortical brain organoid with the right proper cells that we want to study in the lab in a dish. So before I move on, let me just say that if you search now this technology in the media, you're going to end up with names like, oh, they created a mini brain in a dish. So you give the impression that you have like a fully formed brain in a petri dish, which is not true. I'll show you how they look like in a moment. But this is enough for me to highlight the limitations of this technology that I want you to keep in mind. Because we are starting from stem cells, these neurons are still very mature. They are not vascularized. We don't have vases passing through it. So that's a problem. We might not have all the cell types because as an organoid, it means like a small size tissue. So it's a tiny thing. It's not the size of the brain. And we still have problems with reproducibility and translatability of this technology. So lots of things that we need to overcome. And I think most of them, to be honest, we're going to overcome in the next 3 to 5 years. For example, vascularization. I think in the next 3 years, we're going to have a brain organoid that is vascularized. So we can grow them bigger. We can add other pieces of the brain there. All right, so this is our protocol. We have a very nice recipe to make brain organoids. We start with pluripotent stem cells, we neuralize them, we induce their proliferation so they can grow bigger, and then we end up with spheres that are about 0.5 centimeter, and they contain about 5 million neurons in there. This is about the size of a bee brain. And a bee can do lots of things. So you can say that even though it's a very reductionist approach, we have something that might be useful. And this is a postdoc in the lab, and she's holding a plate which contains about 3,000 organoids. Each one of these white dot is one brain organoid with around 5 million cells in there. So we can use this to perform several experiments, and I'll highlight some of these experiments for you in a minute. But just to say that the anatomy is really good. It resembles something that is similar to a fetal human brain. There is a ventricular zone. You have progenitor cells surrounding the ventricle. These progenitor cells migrate and form the cortical plate, which is how the human brain is formed in utero. So we recapitulate all these stages inside the brain. Some people ask me what kind of cells you have. Well, it really depends on what stage you're looking at. I start with single cells. I end up with 5 million cells because the cells replicate. This is a snapshot at 4 months. You have the pool of progenitor cells on top. You have most of neurons being formed. These are excitatory neurons. This is one of the most affected brain type in autism. You have your astrocytes being formed at that stage, and some of the inhibitory neurons start to emerge as well. So as we age the organoids, the diversity of cell populations will actually change as well, making the model closer and closer to the human brain. And that's why we like it, because we can recapitulate very nicely all these stages. And I'll show you one example where this technology was very powerful, and it was another virus outbreak, but this time happens in 2015, 2016, back in Brazil. You might remember the cases of macrocephalic babies that were born in the northeast of the country. So at the time, there was no link between the virus and the defects in the brains of the babies. So to prove causation, what was done was to actually expose the Zika virus to the brain organoid, and we showed that the Zika virus could actually kill the progenitor cells, and so during the migration they would fail, and you have like a very small cortical thickness, which was mimicking exactly what was happening in uterus in those babies. So most importantly than just proving the causation, just 2 years after the outbreak, we were able to use that tool to screen for antiviral drugs that could block the viral replication. And we find one that's called fosbuvir that was initially developed by hepatitis. And so fosbuvir can even actually block what we call vertical transmission from mom to the fetus. If you have a mom that is positive for the virus and takes the drug, the fetus would born without the virus. So this is from causation to treatment in 2 years, which shows that if you have the right tool, you can actually really advance sciences. And this was something that was missing in the hands of the neuroscientists, a really good human model. But autism is not about brain malformations. Actually, very small percentage of autistic individuals have alterations in their brains. Most of the alterations that we see is about networks. And we always wanted to use the organoids to actually measure the networks that was formed during these stages. And the way we do that is by placing the organoids on top of a multi-electrode arrays. So these are electrodes that are just capturing the electrical activity of the organoid, of the tissue, over time. So we can measure that over time and see how it looks like. Scientists were completely skeptical that this would work, and I'll tell you why. Most of the tissues that we had, or the cultures that we had in the past, they were always below what we call the 5 Hertz threshold. This is overall brain activity. So you can see 2D mouse, 2D human cells, or or mouse neurons, they are always below the 5 hertz threshold. And a primate brain fires at about 20 hertz. The mouse brain fires on 18 hertz. So we never could get there. Even if you wait longer, someone waited for 32 weeks, couldn't cross the 5 hertz threshold. So with our protocol, this was the big surprise of the lab. We see that if you wait for 40 weeks or about 9 months of age, which is similar to the human gestation, you actually achieve that 40 hertz with the organoids that we were able to produce in the lab. So that was a big news, and now we can actually study or use that as a model for conditions that affects neurodevelopment, such as neurodevelopment of the network, such as autism. Most importantly, they do that in a very similar way to the human brain. We did that by looking at EEGs, which is the same thing. These are electrodes that you place in your skull, and you can capture the different oscillations. The reason why this was important is that because labs like mine, we are too focused on the basic biology. We like to study genes, we like to study synapses, cells, but it's hard to bridge human cognition. So we'll never be able to do that. But this is something that cognitive neurosciences has been doing for a long time. They place the EEG on people's head, and they correlate the different oscillations that they can get with different human behaviors, including autism. So if you have autism, if you have seizures, your EEGs would be different. And that's what we want to do with the brain organoids. Can we bridge that? And again, I mean, before we start doing that, the only thing that we could do is to look for individual neurons. And I mean, individual neurons will not move you too far. And here's the reason. This is the analogy of a stadium. If I just look for one person, I don't understand what's going on in the entire stadium. Some people are quiet and then they become active. Some neurons are quiet and then they become active. That's cool, but what does it mean? So it means that someone scored a goal, or it means that there is a ola going on in the stadium, right? I only know that if I capture all the electrical activity at the same time, and that's what we were trying to do with these multi-electrode arrays. This is too complex, but just to say that there is a way to do that, and we team up with a colleague of mine here at UCTV, Brad Wojtek, for this study. And long story short, I mean, together with him, we showed that the organoids start to show these oscillations when they are about 4 months of age. By 6 months, they are very stereotyped, and by 8 months, they become highly complex. The activity of the organoids correlates really well with the diversification of cell types that you have in there. All right. We also showed that this is similar to the human neurodevelopment by creating a machine learning algorithm where we train the machine using the EEGs of the preterm baby brains, because we cannot do that in uterus. And then we feed the organoid with, we feed the machine with the data from the organoid. And what we see is a very nice correlation between the trajectory of the human EEG compared to the network of the brain organoid, which again suggests or confirms that the model we have is good enough for us to mimic the oranges or the alterations that we see in uterus, including from people who has mutations in those neurodevelopmental genes, which includes the population of syndromic profound autism. All right, having that model allow us to move forward and now try if the gene therapy approach will actually function in the way that we would expect. We don't want to do that before testing in the lab. I mean, that might have like some side effect, might not work, might not be safe. So the first thing that we want to do is do no harm. So we want to make sure that it's working properly as we had expected before we enroll our kids in this kind of clinical trials. So the first thing that we did was, okay, let's find a cohort of individuals or patients that are very similar to each other, that all have the same mutation, so we can test it. And we started with our friends at the Pitt Hopkins Foundation, and we collected fibroblasts from different individuals carrying mutations in a gene called TCF4. And you can know the gene is mutation by doing genome sequencing. So they all have genome sequencing. They all figured out that they have alterations in TCF4. So they are now classified as Pitt Hopkins kids. And we reprogram these cells and we start the work. So this was all performed with Fabio Papes, who was a visiting scholar in the lab, and he was the one who led the study. So Fabio collected the cells, reprogrammed the cells. These are some of the characteristics that we keep in mind when we try to model the conditions, try to see if we see something similar in our brain organoids. Of course, I mean, there are situations that requires other cell types that we don't have in organoid. For example, constipation. We cannot model yet constipation in a dish. We might be able to do that in the future. So we were looking for more of the intellectual disability alterations in the networks, something like that. And so Fabio was able to create these brain organoids. And one thing that he immediately saw is that the size of the organoids compared to the controls here are the parents of those kids, they are smaller. We can see that they do not grow as you would expect for neurotypical individuals. So the size is about 50% reduced compared to controls, just looking for the diameter. And the expression of TCF4 is also reduced in the patients, as we would expect, because they have mutations, so they're not expressing the full level of TCF4 in there. Also intriguing is that we do see some malformations of the organization of these neurons inside these organoids. For example, the organoids, the normal organoids or the control organoids, they will form rosettes. These are early stages of neurotube formation. You can see those rosettes or these morphologies on the parental organoids, but we barely see that in the Pitt Hopkins-derived organoids. So once we have, and we quantify, for example, SOX2 is a cell type that proliferates in the brain. So you can quantify that, and you show that there is a reduction in the number of SOX2 proliferating cells in these organoids, explaining why they are so tiny, they are so small. All right. So what else can we find out using these organoids? Well, we can look around all the genes that TCF4 might be regulating. TCF4 is a master regulatory gene that controls the expression of downstream genes. We do that by doing like a gene expression profiling. For those who are not neuroscientists, this is just a way to analyze thousands of genes at the same time. And the different colors means that we have more of these genes or less of these genes compared to controls. And by looking at these genes, we can guess what kind of molecular pathways are affected as well. And we are able to create insights about how this gene is actually reducing the number of SOX2 positive cells inside these organoids. And we learned that some of these progenitor cells are actually controlling the identity of cortical neurons that would appear later, a little bit later in life. And these cortical neurons are important for the proper network to be formed. And one of the markers for that is called CTP2. So this is a specific cortical neuron. And you see, again, because we don't have the right proliferation, you end up with a cortical population that is lower on these CTP2 neurons. These neurons are involved on the microcircuitry of the cortex, but also between the communication of the two hemispheres, between the communication of the cortex and other cortical regions. So it's a very important population that might explain several of the clinical symptoms of these patients. So it's very important that we are recapitulating that in a dish. We also confirm these findings in postmortem tissue. We were able to perform the same analysis in one of the postmortem tissues that we had. And we validate that not only the CTP2 population was reduced, but also several of the genes that we were expected to be affected by the lack of TCF4, they were affected as well as in the post-mortem tissue. So that gives us like a confidence that what we have is really something that mimics the TCF4 deficiency, or as we call haploinsufficiency, because one copy is functioning, the other one is not, in the brain cells of those individuals. All right. So we have a model, the model is working. The model now, he recapitulates some of the key parameters that we think is important for TCF4, what we can do. So one strategy, and you probably hear about the CRISPR enzymes. So these are enzymes that you can use to correct genetic mutations, but you can also use these enzymes to stimulate the expression of genes. And I told you that those individuals have one of the normal allele, the normal copy of the gene is still functioning. So what Fabio decided to do was, okay, let's use CRISPR to activate or to overproduce the right copy of the gene. So maybe by doing that, you have more protein inside the cells, and we can rescue that. So he designed the strategy. It requires like some specific guided RNA to the specific regions of the TCF4, and he tested in the protocol of brain organoids. And to our biggest surprise, if you add the CRISPR early on in the development of the brain organoids, you no longer see the macrocephalic phenotype. You basically rescue the population. You rescue the expression of TCF4, but as well as some of the downstream genes. But one of the most, I would say, surprising findings that that he shows, it is that something that we're not expecting to be rescued, which was some of the morphological alterations that he saw in these organoids. For example, here I'm highlighting in magenta there some of the what we call cortical scars. So these are neurons that are just not formed properly in the cortex. But upon the treatment with the CRISPR enzymes, he was able to see that this was gone. So he no longer has these structural defects in these organoids. So these, again, all very good news. The only bad news here is that the CRISPRs are not yet ready for clinical trial. These enzymes are still under development. They create what we call off-targets. So they might activate other genes than the ones that we want. So it's not quite ready yet. And then we thought, what about if we create a second strategy where we just put the correct gene inside the cells without trying to regulate the endogenous one? So it was a little bit tricky to do that because the expression of TCF4 needs to be regulated in the precise amount inside the cell. So he created a very nice strategy that I'll spare the details now. But basically, by manipulating the repeats of this micro E5 box upstream to the promoter region, he was able to get the right amount of TCF4 inside the cells. And then he shows that the virus, he built the virus carrying that gene and shows that the virus was enough to restore the expression in the brain cells and all the downstream genes that regulates TCF4. More importantly, he then used the virus to infect brain organoids already formed from Pitt Hopkins patients, and he started to see back all the alterations that I previously described. First of all, he restored the expression of TCF4 inside the neurons. He was able to restore the structural defects caused by the lack of SOX2 positive cells in there, and the restoration of the CTP2 cortical neuronal population responsible for all this cortical communication. So that was great news. And then we said, all right, what about the function analysis? So he placed the organoids on top of these multi-electrode arrays, and he recorded over time. This is the behavior of the networks of the controls in the first graphics. And in dark blue is the patients, and in light blue is the patients after the viral correction. And you see that we're able to restore some of this network activity. Again, very nicely done. And so I'm glad that this program was picked up by Mazi, which is a company that does clinical trials, and these vectors are now being tested in monkeys for toxicity. Once we clear the toxicity, this would be moved into clinical trials. So we're not expecting any side effects, but the FDA will require that we test those vectors in primates' brains just to avoid any surprise when you're doing the clinical trial. So this is a proof of principle that you can actually correct the gene inside brain organoids and rescue not only the cellular defects, the molecular defects, but also the network defects, giving us confidence that this might actually work in vivo as well in the patient's brain. So in my lab, we're expanding these ideas to other genes. This is another program that we have for CDKL5 deficient syndrome. If you have mutations in CDKL5, you have a very early onset of seizures. When you are like a newborn with 3 to 4 months of age, you start having seizures. So that's dramatically affect your neurodevelopment. And this is due to the hyperexcitability of these networks. The bar in the middle shows how hyperexcitable is the networks compared to the black bar, which is the control in the organoids. And now, if we just replace the gene back into the organoids, you see how can we reduce this hyperexcitability, most likely eliminating the susceptibility to seizures. So this is another program that is moving on for a potential clinical trial. And we think that the earlier we act, the better for these patients. And finally, we have another one for Rett syndrome. This is a gene called MECP2, is an epigenetic gene. We showed that, we and others, we showed that there are many defects associated with alterations in brain organoids that carry that mutation. Most importantly, if you place the organoids from Rett syndrome in these multi-electrode arrays, they will show a deficiency as well. And now if you add the virus, the little bit of green that you have in that organoid in there is the small number of cells that we are able to infect with the AAV. And this is a nice curve showing that as you increase the dose of the virus, you restore the electrical activity of the population. And what I think it's quite important, and we still don't know yet how that works, is that the number of infected cells here is less than 5%. I mean, we are getting like a very low infection, but that's enough to correct the entire network. So that gives me hope that the brain is so plastic that even with just 5% of functional cells, you are able to overcome all the genetic defects. So genetic is not deterministic if you have the proper environment for the neurons to thrive. So that's quite important. So there are many other alterations related to autism. This is just like a sample of the conditions or the genetic alterations that we're doing in the lab. Most of these conditions, we don't have even animal models to do that. So we generate brain organoids. We test for therapeutics. We test if we can find drugs. We test if we can find a gene therapy approach. There are now antisense oligonucleotides, ASOs. You might hear about that. So that's another possibility. And the goal is really to show proof of principle and accelerate, speed up all the clinical trials. And I'm glad to say that over the years, the FDA is being more and more sympathetic to this model, understanding the needs to really push this type of therapies further on. All right. So proof of principle, we have it in our hands and different stages of development now. What are the next steps? There is one major problem with the gene therapy, especially in autism, especially for the syndromic profound autism. It is that not everybody does genome sequencing. This is something that is not like mainstream. Most of the doctors don't even prescribe that because they don't even understand the value of that. To be honest, I mean, even if you have the the gene right now, there is not much you can do, but you can start already personalizing the treatment. Even if you cannot do that, I mean, you might be able to participate in these future clinical trials. And the problem that we are having is to recruit enough patients for these clinical trials because we don't know where they are. I mean, we might have good foundations, such as the Rett syndrome, the CDKL5, the Pitt-Hopkins, that help us to map all the patients in the world. But what about all the other 100 genes related to autism? We still don't have foundations being formed. We still don't know how many of those are affected. We don't know where they are. But the good news is that genome sequencing is getting cheaper. People are having access to that. There are more conscious about the value of that, so people are doing it. So what we decided to do is to create an app called Autism Me. On that app, you just plug your information and you add the genetic alteration that you find in your report. And researchers like me or doctors or even family members, they can search by the characteristics, by the gene mutations, or by the profile, and be able to connect to each other. So we initially thought about doing that for the science, but we realized that there is way more benefits on creating the first social media for autism, actually. That's what it becomes, because there are several stakeholders that might take advantage of a social network like that. So we are seeing like families connecting and exchanging information among them. Some have kids that are older, some had kids that are younger, and being able to connect with the families, what they try, what kind of drugs it work, what kind of treatments they're doing, how they're progressing in life. It's really value for other families. So that's a plus. But also, I mean, all doctors are now finding ways to find other families to point or to ask questions about all these specific genetic alterations, because honestly, we don't have natural history for most of them, so we don't know how they progress, what are the clinical symptoms associated with specific mutations. So that's the value of that. And we created it in a way that you can opt in for research, so it's not automatic. You actually see a value on that, and you decided to participate, and the data is yours, and it's fully protected. So we are running like a pilot experiment in Brazil with my colleagues over there. I think we have about 5,000 people already connected and using the network. The next phase is really the expansion to other countries. And we really hope that this tool will facilitate not only the research, but the interaction among families, among therapeutics, among medical doctors. So we'll see how this goes. I mean, the network would be shaped by us. So we create the network, we can shape it, and we can evolve the way we want. So it's really something that we should take advantage of. All right, so I'm stopping soon just to take comments and questions, but these are the major take-home messages that I hope you keep in mind. Profound autistic individuals are likely the most vulnerable population and might benefit from gene therapies. Again, I mean, in the context of the whole spectrum, this is not for everyone. This is for the ones who actually need it. So the science is working towards that, towards improve the conditions, improve of the symptoms. We might not be able to get to a full cure, but we might improve some of the symptoms. We might eliminate some of the comorbidities with a gene therapy approach. So it acts on the root of the problem. So human brain organoids can be derived from autistic individuals, and they function as a gene therapy approach or as a proof of principle, and might be useful tools for preclinical assays. So we can already test the vector toxicity, some of those fundamental issues that the FDA might get back to us before we move into proper clinical trial. And then finding these individuals with profound autism, with specific mutations in the same genes, it is and continues to be a major challenge. We have some programs for specific genes. And when we ask the help for some of the pharmaceutics, they are refusing because the patients are not organized. They don't know how many patients are there. They don't have a foundation. They don't have natural history. So it's sitting in my lab and it's not moving forward because we don't have that. So we hope that a network like TismoMe might actually help to speed up all these challenges that we have right now. All right. I would like to thank all the members of my lab. Full disclosure, I'm a co-founder of Tismo. Tismo is a genetic company that actually founded TismoMe. And these are members of my lab. I highlight mostly the work from Fabio here, but there are so many people working on these brain organoids right now. And we are in debt to all the families that actually contribute to our research, that donates their time, that comes to visit the lab, donate their fibroblasts, and actually help us to shape the way we are moving with our science here. So thank you very much.
Some questions came in. One from Claudia Villarreal. Will this therapy help a teenager, 16 years old, who suffered from severe hypoxia at birth, as a result at present time has severe speech delay, intellectual disability and autism, ? Genetical study was done, no other reason for his delay was found.
Yeah, so there is several conditions where, first of all, it might not be caused by a genetic alteration.
Right.
And the question is, can we do something about it? Well, I mean, the traditional gene therapy might not work, but again, we might hack some other genes that facilitate the plasticity of the brain to help those that have hypoxia early in life. What I'm learning is that the human brain is way more plastic than we thought before. There are people walking around with just half of the brain and we don't even know because we don't study them, because they don't go to the doctor because they're not complaining. But the brain can do things that we are very surprised. So I trust the plasticity of the human brain, and I think we're going to find how to modulate such a plasticity even later in life. So I truly believe that that's possible.
Hi, my name is Kyle Cabrera. I'm a student at UCSD. I wanted to ask, in going beyond organoids, as far as what you believe the next steps are for this gene therapy treatment, do you personally believe that the cell type, the cell species matters in terms of different regions of the brain? And I ask because in learning about genetics thus far in my major, I've learned that there is evidence suggesting that certain regions of the brain develop slightly differently in utero for people that have autism spectrum disorders, the cuneus, for instance, and that may not necessarily be the case for other species of cells in different regions. Do you feel like the focal targeting of these specific areas and the development of these areas is going to be important going forward?
Yeah, absolutely. And that's why I think we need a better representation of all the cell types in the organoids that we currently don't have at all. But we have the major one, so it's very reductionist, but I think it's a good starting point. And I do think that the species matter. For example, the most used animal model in biomedical research is the mouse. And you have a fully formed mouse brain 20 days. So the human brain takes like 9 months and is still immature. So the species and the cell type and the background, the genetic background do matters. And I think that's why we are failing miserable on developing drugs for the brain because we never had like a good model system. So I have high hopes for the tiny organoids. Yeah.
Here's another question. An anonymous attendee, going back to the beginning of your presentation and from your perspective, is the activism from highly functional Aspergers, autistic individuals, hurting the profound autistic individuals? By the way, that word that you used up there, profound, which is obviously very, well known to this community and so on. I mean, I was these words are turning up in articles now. Yeah. First of all, what do we mean by profound autism?
Yeah, I think it is a tentative or a way to really characterize that population that is most vulnerable. Going back to your first question. And by the way, not everybody agrees on the term profound autism. I think scientists are slowly embracing it because it helps to describe. When I say profound, everybody gets, all right, we know what you're talking about, are the most vulnerable ones. And this is true with all the autism terminology. It doesn't make 100% of the people happy. So some people don't like this term. I think the community is slowly embracing it. We'll see. And if the activism of the high-functioning autism is hurting the ones on the profound autism, and the answer is yes, unfortunately. And that's why I think it's important to contextualize. We already had instances where scientists had been attacked in conferences or bashed in social media because of such terms. And one example is the use of patient as a terminology. These are patient. And then, I mean, if you say that in social media, oh my gosh, people will go after you. And I think it's just, it's either lack of knowledge or lack of contextualization, because I don't think this population, the high functioning, are lack empathy. I think maybe they are just not aware of what's going on. So I think, again, I mean, that's why I encourage all the scientists to contextualize their research and explaining exactly what you're doing. Because once you explain, I mean, and we try with this community. In ATPF, we have several high-functioning autistic individuals, and we talk equally. They fully understand what's going on. I mean, they're not against a treatment for the profound autism, but because we have been working with them for a long time. This is not true in other parts of the country, in other parts of the world, but we need to catch up.
So as a parent of an autistic child, I believe some of the issues came in utero, and there's no history of autism on either side of the family that we can tell for generations. So I'm wondering, so I understand you're coming from the gene aspect of it. What if it's environmental aspect? Or how do you address that versus the genes?
Yeah. So first of all, by not having a history of autism in the family doesn't mean that it's not genetic, because there are mutations that acquire during development. The person, we call them private mutations or de novo mutations. So it's still worth investigating if they don't have like private mutations. But nonetheless, there are certain types of autism or conditions that resemble autism that are caused by environmental factors, and we know a few of them, it's still, I mean, gene therapy might not work because there is no gene involved, but we in the lab are investigating potential drug treatments. So there are maybe some drugs that might actually help the networks to reestablish, help the formation of synapses. One of the first work that we had for Rett syndrome was not on gene therapy, was on finding a drug, and that was IGF-1. And in our tissue culture, IGF-1 works beautifully. So we restore all the synapses that were missing in Rett syndrome. The problem with the clinical trials was that IGF-1, insulin growth factor 1, was a long molecule, was not penetrating the brain. So it failed until a company called Neuroscience created like a short version of that molecule that now penetrates much better in the brain. And I think it's probably the first drug to be approved for Rett syndrome. So there are hope for drug treatment as well.
Round of applause for Alysson. Thank you.
Thank you.